Thanks very much for the kind introduction. Thanks a lot for the very honorable invitation. I'm utterly pleased to come back to Imperial College, where I spent a year, very long time ago. And I have to say, every time I come to London, which is not as often as I'd like to, I pass by Imperial and I look at what's changing, and I see it's continuously growing. My own dormitory has been torn down. Overall, I would say that time has done well for Imperial. I'm not so sure whether the same can be said about myself. But let's start with some more relevant history. In preparing that lecture, which is in honor of Sir Godfrey Hounsfield, I thought it's interesting and appropriate to compare the development of the two imaging modalities, computed tomography and magnetic resonance imaging, which took place at more or less the same time. CT was conceived 4 years earlier, but as you see that the time scales of realization are quite stretched. Actually, MR took quite a bit longer to get basic finally into application, but also the series of key events is kind of mixed in MR. There was the idea of Sir Paul Audebour, which he published then in a first paper, basically demonstrating the method on some test tubes, and then it took a long time and the effort of many people and people in the UK really played an instrumental role in that. Ian Young here and the Aberdeen group in Scotland were key pioneers until finally in 1980 the first commercial instrument came out. CT took off much faster, so from the idea to the commercial instrument, it was only 6 years, but as you see, the first human image was actually acquired before the first paper actually occurred. So the first publication was more of a hindsight of something which has already been achieved. So CT was pretty fast. If you think it's very fast, you should look back to the grandfather of all medical imaging techniques. That's the X-ray technique, where everything happened within 1 single year. So the discovery was not really an idea, it was a more serendipitous observation, occurred in 1895, and already in the next year there were the first commercial instruments on the market, human images were acquired. So we do think that we are living in a fast-paced world, but compared to that, that could never happen nowadays. Regulatory issues alone would take years for something like that to be realized. But this is history. There's a few things I would like to remark, a few side remarks. The original idea was conceived according to the biography I read while Sir Godfrey took a walk in the Burnham Woods. And the second quotation, which I stumbled across, I like very much because it reminded me a lot about some of my own work, is that the mathematics was phenomenal and others had thought about it but dismissed it as not being practical. So that tells you that it's good to know your field, but sometimes knowing too much can actually hinder you to make a big discovery. So certain naive enthusiasm may help to realize dreams which are thought to be not realizable. So nowadays, of course, both CT as well as MR are big business, real big business. So these are some, I should say, rather conservative figures about the MR market, which is about $4 billion per year. Main field strength is 1.5, 3 Tesla is increasing. There's a lot of interest, especially in scientific community, about even higher fields, 70 or more. The range of indications has not changed so much over a year, but the focus has changed. Initially, most of the work was neuro because, like in CT, the head is the most suitable organ to be imaged. But meanwhile, other fields have been addressed and are being addressed. And actually, although MR is now quite mature. In many respects, there's still a lot of very intense and interesting development going on to address new clinical issues, new clinical questions, to get the patient quicker from diagnosis to therapy and to recovery. When preparing the talk, I thought I would not talk so much about the clinical applications, but more about the role of MR in the biomedical science today. And of course, when you are looking at where we are in biomedical science today, it's good to not only look at what you can do with pretty images, but take a bit of broader view of what biomedical science today is about. And it's mainly not about imaging. It's certainly basically driven by molecular biology, by the fantastic insights molecular biology is giving us into the very basic mechanisms which make up living beings, and which can be condensed in the central dogma, which basically says that all the information about living beings is encoded in the genes, and they are then translated into RNA and further on into proteins, and these then assemble to make all the enzymes, complexes, receptors, channels, whatever, which then aggregate to finally form a living being or a human, which has led to some more or less educated discussion about genetic determination, how much of us is really fixed from the beginning because we have a fixed set of genes, and how much is still open to a modification. In an abbreviated version, one can say we are determined by our genes, and actually every day you can read new papers where new discoveries are being made about linking gene to any kind of traits, disposition, even disease, which now has led to the very intense research into personalized medicine, basically trying to make a tailor-made treatment on a patient based on his genetic disposition. And this is a quotation from an editorial of Science: "Genes contain the building plan of all organisms." And I would like to just spend a few minutes to look into that, and it will hopefully become apparent what that has to do with imaging. Of course, in the genes there is not a piece of paper with blue ink on it, so the idea is more a conceptual one, that the genes encode in their genetic language the plan for the final human being. Let's look at some facts about that. One of the big success stories of the genetic determination has been the discovery of the BRCA gene, where BRCA very simply stands for breast cancer, and staggering 60 to 80% of women with malicious mutations of these genes, they develop breast cancer. So this tells you, if you are a woman and you have mutations of this gene, you should be very worried. And this has been recently, I think, also brought to the broader public by Angelina Jolie. But of course, that makes you worried. But if one looks at that from a scientific point of view, one has to state, well, 20 to 40% of women who have that mutation do not develop breast cancer. Why not? Also, one needs to state that in most disease, and that includes breast cancer, more than one gene linked to the mutation is found. And finally, and probably most importantly, 90% of all breast cancer patients do not show this mutation or any other genetic mutation, which puts a little bit, I think, a different spin on the determination about the outcome, which is encoded into genes. And we have to be very careful when we talk about causal relationship versus disposition versus mere correlations, and this needs to be distinguished quite clearly. Another issue and another field which is currently finding a lot of intense research is related to the fact that it has been shown and discovered that actually not all of the gene is expressed all of the time. There are big parts of the gene which currently, to our knowledge, seem to be totally meaningless. And even the meaningful parts, not all of them are expressed, but the part of the gene which are expressed are defined by epigenetics. And that leads to the fact that with one and the same gene, one can get very different outcomes, very different organisms. One example is in bees, so a queen bee and a worker bee are genetically identical. They have all the same mother, the same set of genes, and so they are as identical as can be, but they are very obviously very different organisms. So one can ask, well, what makes this genetic switch? And that's actually a little bit worrisome. It's nothing but nutrition. By nutrition, bees can change their fate from becoming a worker bee or a queen bee. Another and even more straightforward example relates to metamorphosis. So a metamorphosing animal, of course, always has exactly the same genes, but this same set of genes can encode for a caterpillar or a pupa or a butterfly. This still doesn't say that there is something wrong with the idea of genes containing a building plan. It's just a building plan which is more complex than a straightforward interpretation, and it should give us hope if you feel like an ugly butterfly crawling on the earth. Your genes may also contain the possibility that eventually you will be a butterfly flying in the sun. Let's take a look on the other way around and take a look at two genetically very different specimens. Or we can make it even more difficult and look at two specimens which are even more different, like this. And actually, the genetic difference between men and women is greater than between men and chimpanzees, and I won't further comment on that. And it's not just that the primary sexual organs are encoded and make up for that 2% difference. These 2% are all over the gene. So there's definitely a genetic difference, a marked genetic difference, which should be easy to detect. And actually, biometrics is nothing about the science how different individuals can be distinguished. And this is taken from a recent publication with all the possible means to distinguish individuals. And you see, sex and gender do not enter there. So they don't seem to be very good discriminating traits. And actually, you can take any kind of measure you want, superficial, behavioral, biometrical, physiological measure, and you will always find there are some group differences. Males in general are taller than women, but of course there are small males and there are large women. Males have deeper voices, but of course there are males with high voices and women with low voices. So no single trait can be unambiguously used to distinguish men and women, but we don't have a problem to make that distinction. For once, we immediately make a combination of traits, but on the other hand, we make a point of making visible whether we are a man or a woman, and we can be easily fooled that way. And that's actually an interesting recent book on that topic, dealing with the possibility to distinguish gender and race and others. So, identical genes may lead to different phenotypes, as we have seen. Different genes may have at least similar phenotypes. So what does that tell us about the notion that genes contain a blueprint of organism? Well, I think it's not a very good metaphor for what genes are doing. Genes are not really a blueprint for building an organism. I think it's better to relate them something like a manual, which also relates much closer to the way genes are acting. What are genes? They are sitting in the nuclei of cells, and then by signaling molecules, they are basically interrogated, and then they react to that, and by expressing certain molecules, they tell the cell what to do and how to react to some challenge. Also, the final outcome of development is not written in the genes. It's a process which is written in the genes, and we see that when something goes wrong during development, it's not corrected for. When you build a house and the worker drops some piece of the wall, you just reconstruct it because you have a blueprint. You build the house at the end as it should be. This is not the case for genes. Genes determine the process, not the outcome. Now, is that all semantics, and what does it have to do with imaging? Well, I think the big difference, a big conceptual difference, is that if you want to understand a manual, you have to understand the context. The manual in itself, what it says, can have different meanings, can be meaningless. It only gets meaning from the context. And here's some example of some experiments, some readings which are taken out of context of something we want to get knowledgeable about. And if you imagine that these are different scientific groups working to detect what it is, what is behind all these measurements, there may be very heated discussions between the ones working on with that technique and the ones which are looking at that. It may never really be figured out what it is all about, whereas if you have a look at the context, it immediately becomes apparent what we are looking at, and that is what imaging is about. Imaging is, to put it in a somewhat awkward and indirect way, it's nothing but a spatially distributed measurement, and that means images give context. This is not just some nice philosophical ramification. I just want to show you one example where this is currently becoming more and more relevant and apparent, and that is in oncology, where recently a lot of discussion has been arisen from the fact that tumors are not really homogeneous. So if you want to identify and treat a tumor, it doesn't help you to just take a single biopsy. So these are just points from a tumor where biopsies are being taken without talking about the details. This is a grading, so some parts are very low grading, some parts are very high grading. This is a genetic profile of these different biopsies, and it shows that there's a lot of mutations going on. So the gray parts are the ones which change during the development of the tumor, and only a smaller fraction stays the same. And that means if you just make a single point-like measurement of the tumor, you will probably not really get to a treatment of the tumor as a whole. You may treat the patient for just this one clone, which may in the end mean nothing with respect to recovery. So one really needs to get the full picture in order to develop a sensible diagnosis and also sensible treatment of that. So life sciences today are really living on two sides of a still very deep canyon. There's on one side, there's molecular biology with all this fantastic insight into the molecular mechanisms behind living beings. And on the other side, there is systemic life science, which look more on an organ level or on a physiology level, and where also a lot of things and a lot of insight can be gathered and gained. And currently, we are living in very interesting times, because now molecular biology is a point where it goes out of test tubes and really becomes applicable to humans. And then it has to face the challenges that humans are not a homogeneous solution. Humans, any living organism, is a very complex layered structure with many different layers. And in order to really make progress, eventually and finally, we have to bridge that gap and bring the insight and the two communities together to really make, to solve the problems we are facing with healthcare today, and also to learn more about how organisms work. So what has MR to do with that? Since I didn't expect that all of you are MR specialists, just a few slides to tell you what MR is about. The signal source of MR are spins which are located in the nucleus, which sits deep inside the electron cloud. And all the chemistry and biochemistry, everything which makes life interesting is really taken care of by interactions of the electrons. The nuclei, they are deep inside, they are far away, they have mass, they have a positive charge to keep the electron from flying around, and they have this strange property called spin, which means they are magnetic and they spin around. How can we measure these? Well, if we bring these nuclei into an external magnetic field, they will tend to align with the field, but since the interaction is so weak, the magnetic moment is so weak, there will be some and a majority which will be aligned with the field, but there will be nearly the same amount which will just tumble around and being aligned in the opposite direction. And what we can observe and what makes up the observable magnetization is the sum of the excess of the ones which are aligned, which is about 1 part per million. So 1 in a million more goes along the field than opposite the field at the field strengths we're working on today. That's still a very weak interaction, so if you go in front of a magnet, you don't feel anything. You don't feel the attractive force. Can be measured with special techniques, the force, but the way to measure that in order to use that for imaging is a different one, and it's one which makes use of the principle of resonance, which you can very easily mimic by just ringing a bell in front of a row of glasses, and if you ring the bell, exactly the one glass which has the same resonance frequency as the bell will start to vibrate. And that's basically the way we measure MR. The difference being that for the bell we use pressure waves, for MR we use radio frequency waves, and for the bell we talk about 100 to 1000 hertz, whereas for MR typically we talk about 10 to 400 megahertz. And from this analogy to the bell, we can also immediately perceive that with this experiment we can not only identify where there's something ringing with the same frequency, we can also immediately detect something about the quality. A crystal glass will ring much longer than a dirty, cheap glass from the warehouse. Organ pipe will also resonate, but with a different sound. And this also applies to MR and tells us with MR we cannot just detect, but we also can say something about the quality of the spins. The only thing we are still missing is how to do an image with MR. With 10 to 400 megahertz, we cannot do an optical image because the wavelength is several meters. So if we would use some kind of optics to focus the radio waves, we could maybe figure out whether the patient is still in the hospital, but we could never make an image from the inside of the patient. So an image is produced in an indirect way, again making use of this proportionality between the resonance frequency and the field. So if the field is constant all across the body, all the spins, so to speak, they sing the same tone, whereas if we apply a gradient field which is changing across the body, then spins to the right sing the higher tone and spins to the left sing the lower tone. And this way we can discriminate left from right. We cannot yet make an image. Make an image requires to apply the gradient multiple times in different directions. This is which leads to some of the tedious mathematics which I mentioned at the beginning with respect to CT. I don't want to go deep into, but I think intuitively it's clear this way one can, with sufficient measurements, produce an image. Now, if MR would do nothing but just map out where there are nuclear spins, it would be a boring technique. This is an image which does that. It maps out where there are spins, where the protons, the nuclei which we observe, they are mostly in water in the body, and water is more or less everywhere, and it has more or less the same density everywhere. So images which just map out the water content do not contain a lot of information. But if you remember the comparison with the Bell, actually MR can do more than just map out the spins. It can map out some more properties about the spins, and I will go into that. And suddenly the images start to become interesting. We can now distinguish gray matter from white matter. The ventricles suddenly becomes bright, and we seem to get much closer to anatomy by looking at the images in the light of a different contrast. And if you ask what is the contrast mechanism which makes this distinction, actually it's quite surprising. This is a very short-sighted contrast mechanism. It's something which happens on a nanometer scale. Basically, the contrast reflects how much exchangeable protons are at the surface of macromolecules, proteins, myelin, whatever, and the exchange with the free water is what reduces the resonance time, and this leads to this contrast mechanism which shows up so nicely different organs. And actually, it's at least initially it has been quite astonishing and surprising. Why do we get this clear distinction between gray matter and white matter when we know that histologically all these tissues, they are very heterogeneous? We would not expect that with this heterogeneity of tissues, one would get a clear distinction by such a very simple contrast mechanism. Meanwhile, it has become more and more clear why that is the case, but initially, of course, it was taken as a nice given, as a nice gift together with the possibility to make images. And actually, this contrast mechanism called T2-weighted imaging, which just tells about the decay time, is still today probably the one most important contrast mechanism also in clinical diagnosis, because all kinds of pathological tissues, here this white matter degeneration, but also tumors, inflammation, they show very pronounced on these T2-weighted images. If we go one step in scale larger, and look at a micrometer scale and look what is water doing there. Well, water is not sitting still inside of the cell. Water is diffusing around. And actually, if you look at the diffusion time of water during the typical time of MR measurement, which is a few milliseconds, water encounters many of and explores much of its surroundings. So also the spatial resolution of MR is quite low. We get maybe 1 millimeter resolution. We can use this contrast mechanism to find out something about, what barriers does the water molecules encounter, how is diffusion affected. So we get information on a microscopic scale. Also, the measurement technique is macroscopic. And actually, this has turned out to be an extremely useful contrast mechanism in oncology, because it turns out that tumors, they have two properties which are not well fulfilled by other tissues. For once, they are solid. That's why we can feel them as a very basic diagnosis. But for others, they have very little degree of order, which means that they have a long lifetime. And so making a diffusion-weighted images lets stand out very clearly the metastases. If we look at a standard image, so this is a T2-weighted image, we see a lot of anatomy, a lot of details. We see vessels, we see muscles. We also see these 5 blobs, but it is very hard to base a diagnosis on that. But in the light of this diffusion contrast, suddenly everything becomes much clearer. Diffusion also has a very special application when we look at the diffusion in the brain. The white matter of brain is made up of fibers. The fibers are surrounded by myelin to increase the electrical conductivity. And this myelin sheet also prevents water to freely cross the fibers. So water diffuses much faster along the fibers than across the fibers. So if we measure the directionality of the diffusion, we can use the result to then reconstruct the fiber tracts and get a very nice and clear image of the very complex structure making up the white matter of brain. And we can correlate that then with findings from functional imaging, which I think all of you are familiar with, where one can map out areas of activation and see how these areas are connected morphologically. Just to finish the story about the measuring the mobility as a contrast mechanism, we can also measure macroscopic motion with MR and look at the hemodynamic at the hemodynamics in large or smaller vessels. And David Furman had a workshop with examples from that this afternoon. With MR, we can directly and without having to go to any calibration measure in great spatial and temporal resolution how the blood moves out of the heart and goes around the aortic arch. And this is a healthy volunteer. If we compare that with a patient with atherosclerosis, we immediately see a big difference. The atherosclerotic patient in diastole, so after the blood has been driven downstream, the blood is reflected because of the stiffness of the aorta and goes back several centimeters. And that means if there is, for example, a thrombus which ruptures here, then this thrombotic material can go back and end up finally where the vessels get smaller, which is in the brain, and lead to an infarction. And this observation, which we have made quite early in our 4D flow technique, Michael Markle was the PI on that, has led to very intense research on what risk factors and one has to consider when looking at atherosclerotic patients. So that's just a few contrast mechanisms, and MR has many more. So it's a big part of work of people like myself is to figure out how can we get more information, how can we interrogate the spins, which basically are quite remote from the biological instances. How can we get more information out of that? And we can, with MR, we can look at metabolic fingerprints, we can look at perfusion, we can look at brain function, not directly, but via the change in local oxygenation and much more. And therefore, with MR, we can actually look at, get some information at all scales on the long way from the gene to the final organism. Of course, with MR, we cannot see everything. We just see a very tiny little glimpse on each stage, but it can be, and I hope I can show you some example, very informative to be able to look at not only one level, but at different levels on the way of life. So spins, they don't partake in biochemistry. They are more or less the innocent observers, so we can consider them basically as our secret agents to tell us about what they see at the various level we can observe them. We can do MR on multiple scales also in a more direct way. MR scanners are available for humans, but we can also use one and the same technique to look at animal models. In this way, make a one-to-one correspondence to what we see in humans in much more well-controlled environment. And we can go down to, let's say, micrometer resolution with MR microscopy. We don't reach really at the level where optical imaging is becoming strong, so we need to reconcile that, but it gives a lot of insight that we are able with MR to cover a long range of different levels with one and the same technique. So in the following, I would like to talk a little bit about one area of application we're working in. I have to admit, I know a little bit about spins. I'm not a doctor, so I have made a very strong point over my career that I want to apply MR wherever it is useful. So I don't want to become just a neuroimager or cardioimager or oncoimager. I want to explore whatever can be done. And so we have research groups on all major fields. In the following, I would like to tell a little bit more in more depth about what MR can contribute to the understanding of the brain. As I said initially, MR is a fantastic method to make nice images of the brain. Brain can be nice encapsulated in coils, and it's very minimal. It doesn't move a lot, so we can get a lot of high-resolution images, structural imagings of the brain. But of course, the brain is much more than just anatomical structure. So the brain, again, can be visualized as being a network of networks on very many different scales, from the molecular level to cellular, synaptic level, neuronal units, functional regions, to the systemic level. And we know that one thing which have been known from electrophysiology, this is an observation measurement done by Steve Smith in Oxford, the brain is never addressed. And I mean, we are not only interested in the architecture of the brain, we would like to know what the brain is doing, how is it working. And one observation, one fact we have to state is the brain is always on, no matter whether we are doing something or mean to do nothing. The brain is always on. So this is just reflects the brain activity while a volunteer is lying in the magnet, basically doing nothing. And you see there's a lot of activity going on, and this can be used for very fine-grained parcellation into areas of the brain which continuously talk with each other. fMRI has been around for 20 years, and I don't want to go too much into that, just to state that with fMRI we don't measure directly the basically electrical activity of the brain, but as I've already mentioned, we measure an indirect parameter, the hemodynamic response. But meanwhile, we can do much more than just map out where there's a visual cue processed or where there's a verbal cue processed. So it's just an example of mapping out lesion-associated areas of deficits in a stroke patient from a recent study which we did with our neurology. fMRI has been great and has changed the world of neuroscience, but in the way it has been and still is being done, it's a very static technique. So how do we bring together the insight that the brain is continuously operating with this observation of functional areas? For 10 years now, I've been working on getting more dynamic information into the measurement of the brain. And the original idea was to get away from using MR as an imaging technique and rather use it more like electrophysiological technique. So use MR coils just as signal receptors and then read out one channel per coil, and this way follow in which areas underlying the coils there's something happening. And we see here, if you read out the traces, we see how these areas follow, for example, a visual activation. So it was not really meant to be an imaging method, but doing that, we immediately found out that we actually can measure that as fast as we like. We can acquire the data with megahertz rates, which is physiologically meaningless. So as a second thought, and quite immediate thought, came to the idea that one can invest a little bit of the time savings into doing some additional gradient encoding, as shown before, to improve the spatial resolution while still being much faster than conventional imaging. And actually, after many years, this is an example from our current workhorse experiment based on that. So this is now whole brain imaging, 64 cubed 3 millimeter isotropic resolution, and the whole brain can be sampled in 75 milliseconds. So now we can really dynamically look at what's happening in the brain during tasks or during rest. Now, since fMRI is based on this hemodynamic response, one can ask, why bother and measure something which is inherently slow with a fast technique? Well, the answer becomes immediately apparent when one looks at the frequency spectrum of the signal in the brain. One sees it's not just some noisy trace, but one sees very distinct peaks which can be associated with breathing, with ECG. And ECG, since it's so pulsatile, it's not just at the pulse frequency, but it has lots of higher harmonics. And if we do our standard fMRI experiment, all of these peaks, all of these signals, all of this physiological noise folds back into the observation window and basically decreases the sensitivity of the measurement. Whereas if we measure fast enough, we can discriminate the interesting signal, the brain activity signal, from this physiological noise. And this is just from a recent study where we've shown that if we use that technique, here are signal traces acquired very fast, and we see we have a much higher sampling densities, and we also perceive the pulsatilities of ECG and breathing, whereas if you do a standard EPI, you sample in a more or less random fashion somewhere on these traces, and you just have a higher noise level, which is reflected in a much higher ability to discriminate these resting state networks, for example, than we have with echoplanar imaging. And this means that we can, with this fast technique, within a quite short measurement time already, very reliably detect which networks are on and what are the networks, whereas if we do that in a conventional way, we always need to measure a few minutes, some measure half an hour or an hour, until we can discern these networks, which again makes the technique into a static technique. We can then not really figure out what is happening really over time. So one of my postdocs, Shilai Li, she actually looked at whether there's also some correlated activity going on in higher frequency spectrum, just applying the very same analysis pipeline which we use for the conventional very low frequency networks to different frequency windows, one lying between breathing and ECG and one at higher frequencies. And the result actually showed that we have quite a large number of networks, actually as many different networks at higher frequency than we have at lower frequencies. And here just plotted some networks which by visual inspection seem to be very much correlated with each other, which is actually not very surprising, because this activity is not really sinusoidal. The activity has some kind of a frequency spectrum, which also means there are higher harmonics. But it's not only higher harmonics of the same networks, otherwise the power of these networks would have to be much lower. So there is something going on at higher frequencies which is distinguished from lower frequencies. And from a practical point of view, we can now really look at the brain at work, and we can here at a quite low temporal resolution, but we can increase that. We can really see that these resting state networks are not something static, which is just there in the brain, and it's a continuous communication of different areas, but we have a flux of correlation and even anti-correlation in these networks, which changes with a time scale of about 20 seconds. So this is not directly the electrophysiology. It reflects the hemodynamic response. So it cannot be one-to-one correlated with EEG measurements. For EEG measurements, actually, what we call fast is really in the low frequency bands. But the MR signal is not really just reflecting the theta signal, but the MR signal reflects more like the integrated power irrespective of the frequency. And that means we cannot expect that there's a one-to-one correspondence between networks found by MR and networks found by EEG, which are currently investigated quite a lot. And one sees some similarities between the electrophysiological networks and the MAR networks, but also some distinction. The EEG networks seem to be more distributed, seem to cover larger parts of the brain. So it still needs a lot of work to really figure out what is the correlation between these observations. Also, one has to take other factors into account, like vascular pulsatility, which is known also to be a driving force of brain action. One directly and imminent clinical application of this fast observation has been pioneered by Pierre Levin in my group, and that relates to epilepsy. There's quite a large number, about 40% of epilepsy patients, they do not have a lesion which is visible in imaging, and a large part of them do not respond to treatment by drug. So for them, the way to proceed is to establish where the focus of the epileptic event is, and that is being done with this invasive electrocardiogram. So this is an array of electrodes implanted onto the brain neurosurgically. A patient has to stay in the hospital. In our hospital, it's 12 days, and then from the measurements, one can then figure out which area of the brain has to be surgically removed. It would be, of course, highly attractive to be able to replace this very invasive procedure by a non-invasive measurement, and that's what Pierre was doing. So he built up the device for measuring the EEG together with the fast MR measurement to detect from the EEG the epileptic events, and then to use the MR technique to localize the events. And this is just one result from a study which showed that actually he was able to localize every interictal epileptic event which was observed in the study on so far 33 patients. There are still some issues there. There are issues with false positives. The brain is always at work, so at any instance in time when we start making analysis, we will find areas which show correlated activities. That's, I would say, not a very severe problem. There's also a very interesting observation, and that is that there's a very distinct pattern preceding the epileptic event, and we're currently looking at how that can be used then to, in the end, maybe by some intervention, prevent the epileptic event. So this is a very direct clinical application of something which originally was meant really for neuroscientific studies. So far, these resting state networks, they are correlations, and if you remember at the beginning, I warned against making too much about correlations and dispositions. So the next step, we work together with our Bernstein Center of Computational Neuroscience to figure out the connectivity from these correlations, which takes a lot of work for the computer. So it's a very large, extremely large inverse problem to be solved. So I can, without any further comment, just show you the first results showing networks of areas of the brain which show connected activity. And looking at these networks, of course, one thing we have to do is to see how this relates to known functional networks. But the second thing, and very tempting thing, is we would like to bring that together with what I have already shown, the possibility to map out also the connections of the brain. The challenges there is these nice fiber images. They are basically the result of a mathematical procedure applied to diffusion data. So we need to verify them. We need to find the ground truth. We cannot analyze the brain of our patients in that way. Even if you do that on anatomical samples, it's extremely challenging to map that out. So what we did is we tried to implement the same procedure also on mouse brain, where we then have a more direct access to validation. Don't want to go too much into that. It was a big challenge to really scale the methods down, to really map out also the fibers making up the mouse brain. But actually, in the end, we were successful. And by being able to shrink the spatial resolution now to about 100 micron, we immediately get much closer to the real size of the structures we are looking at, because the axons in the mouse brain are not much smaller than in the human brain. So we have much less fibers inside of the voxels, and the images are immediately suggestive to be much more meaningful than the somewhat abstract images we get from the human brain. And just by visual inspection, we can see there's a lot of correspondence with histological slices. We can do better than that, so we are working together with Karl Silles from Jülich, who is using the polarized light imaging for fiber tracking, and produces images which look deceptively like our MR images, but one has to note that he's looking, because of the limited penetration depth, at only 1 micrometer slices. So we do have to consider that we have much more partial volume effects in our measurement, but still doing the measurement first on the MR on our mice. We then send the mice to Aachen, and then they are interrogated with PLI. We see a lot of correspondence, and we can do quantitative analysis. We see some places where we don't see some structures in the MR, but this can be explained by partial volume effect. There's one big mystery still there, and this is this very pronounced stripes, the polarized light imaging, which you just don't see at MR, and we're trying to figure out where the difference coming from, whether it has to do with the polarized light imaging or something we are missing with MR. We, of course, don't do that just for validating our experiments. We do that to work together with preclinical scientists in some of their application studies. This is one example which I find very impressive. This is a mouse which has been genetically modified to not produce reelin. Reelin is a molecule which is known to be important in the cortical layering of the mouse brain. So in the very early stage, all the neurons are sitting on the base plate and then over evolution, they form out this nice layering structure. And if this reelin is missing, then the layers don't form. And it's actually quite astounding that these mice are still living. They're a bit slow, a bit stump, but their brains seem to work sufficiently well. Well, we were looking at what's happening in the white matter in these mice. Reelin has nothing to do with white matter, and we are quite astounded to see that if you look at the wild-type mouse, the fiber tracts exactly map out what you see in the anatomical atlas, whereas in the gorilla mouse, we see that already quite early on, the fiber tracts seem to deviate from their path. They actually penetrate the whole gray matter, and then they find their still sitting there target neurons there. And that tells us that the development of the brain has to be much more complex and sophisticated than just sniffing along a gradient of some substance like reelin. Another example I want to briefly show, which alludes to the original discussion on how much is really defined by genetics. We did a behavioral study on BARB-C mouse with our psychiatrist department. The BARB-C mouse is actually a mouse model which is used in oncology, but it also has an interesting behavioral phenotype. Therefore, it's used in behavioral studies. And after the first few animals, our partners were very excited because they saw great differences. But after some more animals, and using this technique, which just shortly before had developed, we figured out that at some of these mice, they are missing the corpus callosum, which means the two halves of their brain do not communicate with each other, which of course makes it easy to expect that they will be behaviorally quite different from the other. This has not been known before. It's actually very hard to detect
in standard anatomical images. And these mice are as genetically identical as can be. It's one very special strain. They are from the same litter, but still, in spite of the genetic identity, some of them seem to have this defect and others don't. Second part to get to this connectivity study is to build up the resting state fMRI. In the interest of time, I want to go quite fast through that. So we managed to build up also the resting state network analysis for mouse brain, and we saw the mouse brain. Yeah, well, it has a lot of complexity, so we get 87 different components, so the mouse brain is quite active, and this is our result from a study where, again, one simple, very single gene was knocked out, gene encoding for this mu-opioid receptor gene, which is more or less present all over the brain, but has a role in the reward circuit. And doing the analysis, what we figured out in the end, that this very distinct change of just one single gene actually changes the whole network. There's only two connections which are left unchanged. All the other networks are totally changed by this one genetic manipulation. Coming back to the correlation of functional and morphological connectivity, well, we see some similarities between the functional and the structural connectivities, but yeah, the similarity is not overly great. Actually, that's not all that surprising, because if we look at what we are actually measuring with our nice fiber tracking, is one thing we note that the termination point of these mathematical fibers always seems to be at the top of the gyri. There seems to be no termination at the bottom of the sulci. Does that reflect that there are no nerve endings there? No, it just reflects that the algorithm, when there's a situation where there are U fibers which are abundant, it will just merge these fibers and identify them as one and the same fiber, and the algorithm only finds terminating points at the bottom. So we need to work more on really find out what are the true endings of the fibers. So that's just a few examples. I didn't go into the next level of looking at microscopic slices, which have also recently built up. So there's a 1 millimeter image of a hippocampus of a mouse. So with MR, we stay in this hierarchy of levels. We stay more or less on that side. And that, of course, means if you really want to understand what's going on, we need to correlate our findings with other techniques, and we need to work together with molecular biology, with neurophysiology, with computational neuroscience to really get stuff together. And this is not just something which has to do with sampling more information. Currently, I have a little bit the impression people think that if we just acquire more and more information, eventually we will figure out what is happening. I think there are very many, much more fundamental issues we need to address. Each of the levels is studied very well and is extremely complex. There's a vast gap in complexity between the different scaling orders. And actually, there's no good indication. Actually, there's quite a few indications against that our reductionist approach of understanding will really work for living systems, or at least for the brain. How do we understand things? Well, basically, we map the world, which is, of course, much larger than our brain, onto the brain by building appropriate models, and mathematics is, of course, a very good way to model the world. And yes, we also have some intuitive models which helps us to understand the world. And this works very well for the physical sciences, but there's no guarantee that this also works for the understanding of living organisms. I don't think it's time to give up, but I do think it's time to really consider more fundamental questions and not just acquiring more and more data and then maybe throwing them in a huge computer to finally figure out what's happening. And if that's construed as criticism against the Human Brain Project, that's exactly correct. So let's go back to the central dogma. We have to admit that in this world of genetic expression and building, all the work we have been doing with MR is on this some detail level. So maybe it's not so relevant. I actually do think that there's a lot of very fundamental issues in this some details, and this is driven more by the engineering comparison. I think there's much more to build an airplane than just self-assembly of components and devices. And I do think there are very fundamental questions. One question which I continuously stumble about and wonder about is an organism is highly complex, and we know from biological systems they are very flexible. They never do the same thing twice, not like a machine. So there's a lot of softness and adaptability in there. But in the end, the system is still, or I think because of that, extremely stable, both in function and dysfunction. We don't get some disease by some random breakdown, but also disease comes in syndromes. So why is this extremely long and complex cascade from the molecule to the brain? How can that be stabilized? I don't think this is possible by just looking at a one-way correlation. If you play golf, then if you're a good golfer, you may have a hitting chance of about 30% from that distance. But the brain, it has to work 100% all the time. When you cross the road, it's not enough if you detect 30% of all the cars which are coming. You really have to detect 100%. So how can that be achieved? Well, in the example of golf, it's an easy way. You just tie a ribbon to the ball and you pull it into the hole. But actually, there's not really an indication that such a top-down mechanism exists in living brains. So it's something which is a fundamental challenge to understanding. And as a last example, if we look at how the brain responds to an external stimulus, so for example, in the visual cortex, we get a signal and we make single cell recordings, and then we see that all the single cells, they fire in a very random fashion. They increase on average their firing rate when there's a stimulus coming, but it's totally unpredictable. It's not even Gaussian distributions. It's very strange and very uncorrelated. But the total signal caused by all of the neurons, it's very stable. It's all the same. You can read up in the books that this has this very distinct peak. So how do the neurons, which individually seem to be totally chaotic, how do they work together to get this very stable answer? So I'm at the end, just a few take-home messages. I think it needs more than just more data to understand the mechanisms of life. Science is really built on reproducibility. That has been the core of science. You do an experiment, you repeat it, and it has to have the same results. But biological systems are never exactly reproducible. So how do we build a good science which takes care of that? And finally, don't forget to take a walk in the wood every once in a while. And with that, I'm at the end. I would like to thank you for your patience and attention, and would like to thank all the people contributing to the work, which has been great fun and still is great fun over the times. Thanks a lot.
Thank you. Just a few words that bring you